CD8 positive cytotoxic T cells, or CTLs, are important both for adaptive immunity to intracellular pathogens and tumor surveillance. CTLs function by killing infected or cancerous cells by direct or indirect mechanisms. Such killing typically follows the recognition of foreign antigens presented by the target cells themselves. Such antigens are usually presented in the context of MHC class I molecules. Direct CTL killing requires cell-cell contact and commonly results from the release of cytolytic enzymes such as granzyme B and perforin. Perforin released by the CTL forms pores in the membrane of a juxtaposed target cell, allowing passive inward diffusion of granzyme B, which then triggers apoptosis of the targeted cell. Direct tumor cell killing can also result from an interaction between Fas ligand, expressed by the cytotoxic T cell, and its receptor Fas, which is expressed by the target cell. Ligation of Fas results in target cell apoptosis via a caspase-dependent pathway. In addition to these direct killing mechanisms, cytotoxic T cells can also induce indirect or bystander tumor cell death by secreting cytokines that work at a distance. TNF-alpha secretion, for example, can induce apoptosis in adjacent tumor cells bearing the TNF receptor. The killing of cells, as described, is fulfilled by CTLs that are also categorized as TC1 cells. However, there is now evidence that naive CD8, like CD4 cells, can differentiate into multiple CD8 subsets in addition to TC1 cells. Accordingly, different cytokine milieu can also generate TC2, TC9, TC17, or CD8-positive T regulatory cells. These cells, like the various T helper subsets, express specific transcription factors and produce signature cytokines and effector molecules. Cytotoxic TC1 cells, for example, are generated in the presence of IL-2 and IL-12 and acquire the ability to secrete cytokines such as gamma interferon. Cytotoxic TC1 cells also produce molecules involved in cytolysis such as granzyme B and perforin. Transcription factors implicated in TC1 generation include TBET, BLIMP1, and IRF4. TC2 cells, in contrast, are induced in the presence of IL-4 and produce IL-5 and IL-13. TC2 cells are generated in response to the lineage-specific transcription factor GATA3. Unlike TC1 cells, TC2 cells exhibit only low levels of cytotoxicity and have been shown to contribute to the development of allergies and autoimmunity. The T cell receptor, or TCR, is part of a complex of integral membrane proteins responsible for the activation of T cells upon antigen recognition. The TCR complex is a defining characteristic of the T cell lineage, and its components are frequently used as T cell markers. The T cell receptor is most commonly expressed as an alpha-beta chain heterodimer. Each chain of this heterodimer has a highly variable extracellular domain region that is responsible for the recognition of antigen presented in the context of MHC molecules. The TCR does not function in isolation, but is associated with a complex of 4 distinct invariant transmembrane proteins called CD3, or the T3 complex. These proteins assemble and function as 3 pairs of dimers, consisting of epsilon and gamma chains, epsilon and delta chains, or 2 zeta chains. The function of CD3 is to transduce intracellular signaling cascades in response to antigen recognition by the T cell receptor. Optimal signaling in response to antigen recognition is further dependent upon the recruitment of either CD4 or CD8 co-receptors. Ligation of the T cell receptor by peptide MHC complexes, presented by antigen-presenting cells, results in the recruitment and co-clustering of either CD4 molecules on T helper cells or CD8 molecules on cytotoxic T cells. This recruitment results in the formation of ternary complexes between the TCR, MHC molecules, and either CD4 or CD8. The introduction of CD4 or CD8 into the TCR complex subsequently potentiates signaling via CD3. Ultimately, the signaling cascades initiated can result in a wide variety of outcomes, including T cell proliferation, differentiation, and cytokine production.